Hi everyone, welcome to our Lab 3 overview. And so today we're going to do a little PowerPoint and then I will talk to you all a little bit about what you're going to do for Lab 3. So in blood bank, the testing that we do is really sequential, and so we continue to build on the tests that we know how to do, and we add something new to them. And so this week, or I should say for Lab 3, we're going to build on our ABO forward and reverse typing, and we're going to concentrate with this lab a little bit on week D testing and RH control. So if you've been able to listen to the lectures, the lecture on the RH blood group system, we talked a lot about week D and the RH control, and so this lab is really going to be an additional reference to back up the information that we talked about then. So when we're doing this typing in the lab via the tube testing method, we're going to label 4 tubes for our forward typing, and so we have our FA, our FB, RHD, and RH control. We're going to eliminate FAB today, so 4 tubes. And then we're going to need 2 tubes for our reverse typing, so our reverse A cells and our reverse B cells. And so with the tube typing procedure, remember our rule, we always want to add our serum source first, whether that be an anti-serum or the patient's serum or plasma. And remember, when we're using patient serum or plasma, we double up because patients are not as efficient as making antibody as a company would be in making anti-serum. And then we're secondly going to add the cell source. So the cell source is either going to be our patient's 2 to 5% red cell suspension that we've made, or it's going to be our reagent A1 or B cells. Okay, so let's just do a little review of the procedure. So for step 1, we're going to add our serum source. So our FA tube is going to get 1 drop of the anti-A. Our FB is going to get 1 drop of anti-B. RHD is going to get 1 drop of anti-D. And the RH control tube is going to get 1 drop of something called RH control. This is basically the diluent that goes into suspending anti-D. Okay, so it's just the diluent alone. For our reverse typing, remember it's always patient's serum or plasma that is the source, so we're going to double up with 2 drops into each one of these tubes. We're going to follow up and add our antigen source. So with forward typing, remember we're trying to determine which antigens, if any, the patient has on their red cells, so we're going to add 1 drop of the 2 to 5% red cell suspension on this patient into each one of these tubes, all 4. Get the patient. Okay. Reverse typing, our RA, this is the known, 1 drop of A1 red cells, and reverse B gets 1 drop of the reverse B. So once we've added everything, we give them a little brief mix. We place them balanced in our centrifuge. We're going to centrifuge for 15 seconds at 3,500 RPMs. And when we are finished, we're going to take those tubes out and put them back in order in our rack, the same way before reading. So the rack organization, we want to organize our rack the same way every time. I don't have a picture this week, unfortunately, of this, but we want to have similar tubes on different patients organized in the same column. So the first tube should be our patient's original tube with the red cells, and they would all be going, that way. Then that patient's 2 to 5% red cells, and so the next patient's 2 to 5% red cells would be behind that one, and so on and so forth. Our FA tubes, our FB tubes, RHD tubes, RH control tubes, and then the patient plasma and reverse A and reverse B. Okay? So organizing a rack in the same way every time eliminates problems or mistakes from happening. So we want to continually organize the same way each time. And so a lot of these pictures are going to look familiar. So when we're reading, remember with tube testing, I say button down in the mirror. And so here you can see the button is down in the mirror, and you can see that with some gentle mixing, this is our 2+ reaction. we would have to keep suspending that button until it was fully off the bottom, and with negatives, it's going to be a smooth suspension. So we've looked at this picture a lot as well. So 4+, 1 clump, 3+, maybe 2 or 3 big clumps, 2+, smaller clumps with a clear background, 1+, the clumps are smaller and the background is now looking cloudy, and a negative smooth suspension. So remember our expected ABO forward and reverse typing reactions. You can't hear this too many times, guys. So remember, forward testing, we're using known anti-sera to tell us which antigens are present on that patient's red cells, if any. Reverse typing, we're using the patient's plasma with known red cell antigens to tell us which ABO antibodies the person has. And we always have to remember LynnC rule, which states that patients have naturally occurring ABO antibodies to the antigens that are not present on their own red cells. So here we see forward O. O has no A or B antigens, and so we see 4+ reactions with A cells and B cells detecting anti-A and anti-B in the patient. When we look at type A, we see a 4+ reaction here with anti-A, nothing with anti-B, so just the A antigen. We expect them to have anti-B, which we see detected here with our type B red cells. So the anti-B is reacting with the B antigen on the red cells, and this is a 4+. There is no anti-A, this is negative, and that's what we would expect. With type B, we see no reaction with anti-A. They lack A antigen, but a 4+ with anti-B, and so we suspect that someone who's type B is going to have naturally occurring anti-A, and so our anti-A in the patient is reacting with the A antigen on our known cells, 4+, and they do not have anti-B. Last blood group, AB, we see 4+ reactions with anti-A and anti-B because they have both antigens, and since they have both, that means we expect them to have neither anti-A nor anti-B, and we see 2 negative reactions here in our reverse typing. So let's move on now and talk a little bit more about weak D testing. Weak D testing is an application of the indirect antiglobulin test. And so basically, we perform the weak D test on any patient who has initial negative with that RHD tube. If we are testing the RH control tube with that patient, we carry the RH control tube along with the weak D test as well. So we're going to take both the negative RHD tube and its corresponding RH control tube and carry them both through the steps. Now, here's the thing. If someone tests positive in the RHD tube initially with a negative RH control, do we need to do the weak D test? The answer is no. That person is RH positive and they're not reacting with the diluent in the RH control, so no, there's no need to do a weak D test on that person because we've already detected that they have the D antigen. So this is just going to be done on our patients with RHD negative tube, and we carry the RH control tube along for the ride. So here are our steps. The first thing we're going to do is we're going to take those 2 tubes and we're going to place them in our 37-degree heating block for 15 to 30 minutes. This is going to allow for the first stage sensitization for antibody and antigen to come together. Maybe they need a little bit more time. Okay? After that step, we are going to take these 2 tubes and we're going to wash them 3 times with our physiologic saline. Why? Because we're getting ready to add AHG, and we always have to wash 3 to 5 times before we add AHG to prevent neutralization and to get rid of any unbound proteins, antibodies or complement that are present in our tube. In our student lab, we would be using an automated cell washer for this step, and one of the video links that has to go along with lab today is going to be about using an automated cell washer. It makes the process very quick and easy. So once we've washed our 2 tubes with the saline and we've removed any unbound proteins, we are now going to add 2 drops of our anti-human globulin reagent. Most labs are going to use polyspecific, so it's going to be 2 drops into the RHD tube and 2 drops into the RH control tube. We mix, we centrifuge for 15 seconds, and we're going to use our viewing lamp in the same way to read the reactions. So when we're reading our reactions, we want to grade any positives. If we see a positive in the RHD tube and a negative in the RH control tube, guess what? We're done. It means this person has weak D present. And so they would be considered RH positive. That's how we would interpret them. And so remember, weak D testing, weak D basically can result from inheritance of the D allele with respect to C and E. And it can result in weaker expression of the D antigen. That's one of the times we can see it. Okay. Now, if, however, both the D tube and the RH control are still negative, as they were initially, we are not finished. Okay, we have to move on to step 4, as we would in any antiglobulin test where we get negative reactions at this step. We have to move on to check cells. So remember, check cells are reagent red cells type O, which happen to be coated with IgG, and they come that way from the manufacturer. We're going to add 1 drop of the check cells into RHD and 1 drop into RH control. And once again, we're going to give them a little mix and centrifuge them for 15 seconds. After that time, once again, we're going to read and grade reactions. We expect the check cells to be positive at this point. What the check cells are doing, look at them, they have IgG coated around the cell. So when they come into contact with the AHG that's in that test tube, this anti-IgG should agglutinate those check cells, and it's basically proving that our reagent was added, everything was washed appropriately, but what it means is for the patient that this is truly a negative result. Okay, and we would expect both RHD and the RH control tube to be negative. Okay, what happens if our check cells don't agglutinate? It means our procedure is faulty. Maybe we forgot to add AHG. Maybe our AHG wasn't reactive anymore. Maybe we failed to wash appropriately and there were some proteins present in the tube that neutralized our AHG. It could be any of those things, but negative check cells always means my procedure is faulty and I need to repeat it. So you've seen this picture before. Here we see our polyspecific anti-IgG, which contains both, polyspecific anti-human globulin, which contains both anti-IgG and C3. We see monospecific anti-IgG. This one happens to be pigmented, and the reason it's pigmented is so that we can see when it's been added to the tubes. And then we have our check cells, and you can see here IgG coated red cells. Okay. Okay, so let's move on to the next slide. So just a little bit of a review of what happens when we add check cells to our tube. Remember, in that tube, we have antibody against IgG. The check cells come from the company coated with IgG surrounding the cell. So when we add the check cells into the tube where we have our patient's red cells that did not get linked up with the anti-human globulin, we see that it has 2 binding sites available. So when the check cells are added, this antibody says, hey, there's IgG, there's my target. And it happens to be on a red cell, so we're going to see that agglutination. Okay? So we always expect the check cells to agglutinate. Remember, they can be called Kuhn's control cells, after Kuhn's, who was the scientist who came up with this idea. But what these check cells mean for the patient is that the patient has a negative result. So for instance, our check cells agglutinated, it means our AHG was added, it functioned properly, but in this case, the patient would be weak D negative or RH negative. So here's a summary slide, typing for D antigen. And so basically here we have our patient cells and anti-D in the tube. We spin and read, okay? If with our initial testing, the person was D positive, fine, we stop. There's no need to do any weak D testing. If, however, the tube was negative, a smooth suspension, we're going to move on to the weak D test, which is an application of the indirect antibody test. So as we discussed, this entails incubating the tubes for 15 to 30 minutes first. After they're incubated, we're going to wash them 3 times. Okay, get it into your head that before we add anti-human globulin in any test, we must wash the cells 3 times. So we wash them 3 times. We add our drop of anti-human globulin, spin and read. If now it's positive, we're going to report this person as D positive. Weak D means Rh positive. If they're still negative, we're going to run our check cells, and assuming our check cells agglutinate, then we're going to report out the patient as D negative. So one more thing before I move away from this slide, just remember the implications of someone who is weak D. We would absolutely, if we were working at a blood donor center, need to identify someone who's weak D positive. Because if we fail to do so, and we call that person Rh negative because we missed weak D, what's going to happen? Their blood is going to be labeled as Rh negative. Their cells could be given to an Rh negative recipient, but remember, they really have D, okay, in a weak variety. That Rh negative recipient can now be sensitized, exposed to the D antigen, and make antibodies against it. Okay, so we don't want to miss it in that respect. So here I have an example of some different results from weak D testing, which I'd like to review with you, and hopefully it's going to make sense why we did certain things and what they mean. So let's look at patient number 1 here. Okay, we see forward A negative, forward B negative. We see reverse A 4+, and reverse B 4+. So based on forward and reverse typing, this patient definitely looks like type O. When we look at the D tube, however, and the Rh control, we see that they have initially tested negative. So what that means is we would want to move forward with weak D testing because of this negative RhD. And so what we did was we moved forward with weak D testing. Remember, we're going to carry along the Rh control as well. So we weak D tested the RhD tube and the Rh control tube that goes along with it. So let's see what happened. So we incubated for 15 to 30 minutes. We washed the cells 3 times. We added AHG, and we see here that in this patient, after AHG, we got a negative result in both the RhD tube and in the Rh control tube. So we're not done. Okay, we need to add our check cells to ensure that the technique was carried out properly and that our AHG functioned appropriately. Check cells were added into both tubes. They were recentrifuged in red, and we can see here that the check cells both agglutinated at 1+, and you see my little check mark. So that means that the check cells are proving that our procedure is not faulty. AHG was added. Okay, what does this mean for the patient? It means this patient is truly negative. So we're going to call them O negative, the absolute universal red cell donor. Let's move on to our second patient. We could see with forward typing, this patient is testing 4+ with FA and FB. And so based on forward typing, they're looking like type AB. We expect them to be negative with both reverse A and reverse B and lack both antibodies since they possess both antigens, and we see that that happened. So we could fill this in as AB, okay? When we look at the initial RhD and Rh control, we see once again that they are both negative. And so remember, based on this result, The RHD result being negative, we're going to take both of these tubes and we're going to proceed to the week D testing. Same procedure. Put both those tubes in a 37-degree incubator for 15 to 30 minutes. After that incubation time, wash 3 times with saline. Add our AHG to each tube. And lo and behold, what do we see? In the RHD tube, we're seeing 1+ agglutination. This can be a very weak reaction, and that's okay. In the weak D RH control tube, we're seeing a negative reaction. So with the indirect antiglobulin test, remember that when we get a positive reaction with AHG, there's no need to do check cells. Okay, so what we're going to do is only do the check cells on the negative RH control tube, and we see that they agglutinated 1+. Okay, so what does this mean for our patient? Okay, it means our patient really does have the weak D antigen, and so we're going to call them AB positive. Okay, so let's move on to the third example. So with this patient, we see a negative result with FA, 4+ with FB, and once again, negative with the RHD. Okay, so first of all, forward type, what does this patient look like? B antigen, they look like type B. So we expect them to have a strong reaction in the reverse A tube and negative in the reverse B, which we do. Okay, so they're typing as a type B. Now, because of this negative result in the RHD tube, once again, we're going to carry the RHD tube through week D testing, and we're going to bring along the RH control. So what do we see? So with our RHD tube, okay, after addition of AHG, we got a negative result here, and also with the RH control tube, which means we need to move on to our check cells. So we add a drop of check cells into the same tubes, centrifuge again. We expect to see agglutination like we did here in our first patient, but this time we're seeing negative results with check cells in both the RHD and in just the RHD tube. Okay, so the check cells for some reason worked okay in the RH control tube. So what does this mean? This is bad news. Okay, because these check cells did not agglutinate, it means our procedure is faulty. Did we accidentally miss adding the AHG to this tube? Was the AHG faulty in some way, or was there a problem when we were washing this tube? We don't know, but what it means is we cannot interpret at this time the RHD, and so we basically would need to repeat the week D test. So set up 2 new tubes with the RHD and the RH control, add a drop of anti-D and a drop of RH control, 1 drop of patient 3 to 5%, 1 drop of patient 3 to 5%, incubate, go through the whole step again, wash 3 times, and hopefully, the next time when we do, this test, we're going to get a reaction with our check cells. So because the check cells did not react, we really can't interpret what happened in this tube. Okay. So our last example, we have a 4+ FA, negative FB. So forward typing, this patient looks like they're type A. So we expect them to have a strong reaction in reverse B tube. Oh, and I forgot to put it in there. Sorry. So I'll have to add that to the PowerPoint that's loaded on Canvas. I made a mistake there. And basically, we see 2+ with RHD. Whoa-ho. So initially, we're just looking at this RHD. We can say, well, this looks like this person's A positive. However, when we look at the RH control tube, we see 2+ agglutination happening here as well. And that's bad news. So what that means is we cannot interpret the RHD on this person. And it's really because we're getting cross reaction in the RH control, which is simply just the diluent. Okay, so we don't know what's causing the agglutination in this tube. It's really because they have D antigen, or is there something else going on there? Okay, so can't interpret here. In this case, we'd want to repeat, maybe with different anti-D, and maybe a low protein anti-D and see if everything works out okay. If not, we'd have to move forward and do some additional investigation. So hopefully this all makes sense. So the last slide of this presentation, I wanted to just review something we talked about a while ago when we talked about anti-human globulin reagents, and this is neutralization of our reagent. Okay, so neutralization is a big reason why we get false negative reactions in the anti-human globulin test. And so you can't hear this too many times. Hopefully you remember it from lectures 2 and 3. So the rule of thumb is anytime we're getting ready to add AHG, we need to wash our tubes 3 times with saline to remove unbound proteins, which could mean junk antibodies that could be present in the sample. Okay? Failure to wash appropriately, let's look at this tube. Okay? So in this tube, we have an antibody that's attached to a red cell and one here, but there's no lattice formation, the second stage of agglutination, and so we're unable to see this, which is why we've moved on to the antiglobulin test. So unfortunately for us, we did not wash properly, and so there's this free junk antibody that's just floating around in the tube with our immune complexes as well. So when we add our AHG, our anti-IgG, which I have designated as the brown antibody, when the AHG hits this tube, it's anybody's guess what it's going to bind to, okay, because its target is IgG. So we have IgG that's floating around free, and we have IgG that's bound to red cells. Whichever one it comes into contact first is what it's going to bind. In this case, we could see that it came into contact with the junk antibody and bound to it. So if its binding sites are taken up by binding to the junk that we failed to wash away, there are no binding sites available to latch onto these IgGs. And so this looks like a false, or this looks like a negative result. It's a false negative result because we're failing to detect this sensitization, which has already occurred. Why are we not seeing agglutination? This junk antibody is not affixed to any red cell, so it's going to look like a negative in the tube. So this is neutralization, and remember, it is a primary reason for false negative anti-human globulin tests. Okay, so I'm going to stop. I'm going to pause. Okay, so I just wanted to show you guys some additional things that are important for today. So when you go to our Blood Bank Lab 3 page, PowerPoint, the recording link will be here. There are some supplemental lab videos that everyone should look at today by clicking on the link. Let's see what happens with mine. Okay, if it can show you. Yep. So the links today, what you should be looking at. We have a video today of ABO forward and reverse typing, including week D testing. And then we also have a link that shows the use of an automated cell washer. So you really do want to look at both of those. There is a week D procedure here. If you would like to see it, you could look at it, but it basically outlines a lot of the things that I included in the PowerPoint, but this is a Word document if you would like to look at that. And then you absolutely want to download the lab report by clicking on this link, okay? And then you're going to use this link to take you to, let's see if it'll keep my share, okay, the Nobel Prize blood typing game, okay? So download the report. Let me stop the share and show you before you play the game, like last time, okay? So when you download the report, this is what it looks like. And so here are the directions. You could click on the link, as I showed you. on our Lab 3 page. Remember, you are going to need to have Flash activated to do this activity. You can go to the link and you're going to play the blood typing game. And you're going to do quick random patients today. And I'll show you with my account. And you want to have this file open as you go. So once you start playing today, you're going to type 3 patients and then select units that would be acceptable for transfusion. So when you do your 3 patients, it's going to add the reagents to the 3 tubes, and basically you're going to just tell me positive or negative for each tube and what type you think it is. And then when you're playing the game, the units that you select for transfusion. So you're going to do that playing the random game for 3 patients and type your results in there. When you are finished with that, the second part of the lab report for today has to do with an activity that's similar to what we just did together. Okay, so I have 5 patients listed here. I want you to evaluate forward typing results, reverse typing results, tell me the ABO type, and then look at the RH testing, and if there's weak D testing, use that as well. If you can interpret, tell me the RH type. If you can't, tell me why, okay? And then there are a few questions related to some of the things that I talked about in the presentation today. Okay, so you want to look at that. I don't think you guys saw any of it. Oh. So sorry, guys. I don't think my screen share worked. There it is. So anyway, here is the report. Go to the blood typing game, put in your results for the 3 tubes, your type, units you use for transfusion for your 3 patients, and then an activity here that is similar to what we did in the PowerPoint together. Okay, and then some additional questions. When you're done filling all of this in, as you usually do, save it to your computer, and then you will upload this as a file upload when you are done. So let me stop this share, go to a different share, the blood typing game, maybe you remember it, here it is. Okay, so when you are in the blood typing game and you click on play the typing game, it's going to load. I don't like the sound. Click proceed. You may have to enter your email at this point. And so when we did this game last week, we did the mission-based game. This week we're going to do the quick random game. And so basically it's going to show you 3 patients. Okay, so you draw the syringe, fill in your tubes. Okay, so what do I think this patient is? I'm going to also click this on my worksheet. So I think this person is B negative. Okay, and I'm right. But guess what? What am I going to do? Bear with me. I'm also going to have my lab report open. So in my case, patient 1, I'm going to type in here negative, positive, negative, B negative. Then I'll move on to the units for transfusion. Okay, so I move on. And some of them may ask, you know, the number of units that it asks for are a little bit different. So this time they're asking for 2 units. So what of these would be acceptable to give to someone who's B negative? Well, no, 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 yes. I drag B negative. I see that I don't have another B negative, so I have to keep clicking forward. I can give O negative, that's universal donor, right? Okay, so basically those are the 2 units that I used, okay? Before I move on to my next patients, I'm going to go to my worksheet, and in this case I'm going to say B neg and O neg, okay? And then go on and fill in my results for the next 2 that I type, okay? And then, remember to evaluate the results I'm giving you related to week D testing and answer some questions. Okay, so that will do it for today. Good luck, everybody, and I will talk to you next time.